Hi everyone, and welcome to the last part of chapter 16 that you're going to learn. Now, chapter 16, specifically part 3b, is about mutations. But before we go into that, let me do a little summary of what's going on here. Now, in the beginning of last week, we learned monohybrid and dihybrid inheritance, and this is the classical examples you have learned in O levels or SPM. Yeah, what is it? Monohybrid is when one gene with two alleles, very normal dominant recessive type alleles, codes for one trait. So the example we learned is tall or short, right? Which is one trait, two types. Now, F2 ratio, F2 is the product of crossing F1 and F1, right? And F1 are heterozygotes. Both of them are. Yeah, and if you do that, you get a 3:1 ratio. Now, in dihybrid cross, we really are talking about two genes on different chromosomes, two alleles each. Again, the normal dominant recessive type ones. But you can see here the F2 ratio differs because instead of one gene, we are talking about two genes. Okay, two genes. So F1 cross with F1, okay, which are heterozygous for both genes, would result in a 9:3:1 ratio. And you should be familiar with this by now because, well, you drew a lot of genetic diagrams last week. Okay, anyways, the next one we learned was sex-linked, specifically X-linked, X-linked inheritance. And this is the idea that the gene is on the X chromosome but is absent on Y. Now, this really depends, okay, F2 ratio depends on whether the allele is recessive or dominant, okay, and yeah, whether the dad is the one with the X-link sort of gene or is it the mom, okay, so this depends. Number 4, we learn codominance. Codominance is when there's one gene, two alleles, but those two alleles are fully expressed in heterozygotes. Okay, so there is another phenotype other than just dominant phenotype or recessive phenotype. So the F2 ratio here is 1:1. Okay, again, the F2 ratio is when F1 is crossed with F1 and produce F2. Okay. So, yeah. Next, multiple alleles. Multiple alleles means there's one gene, but there's multiple alleles for that gene. Means more than two alleles. So maybe 3 or 4. And this really depends, the F2 ratio really depends on what the conditions are, how the relationship of those alleles are. It might be that those maybe 3 or 4 alleles are in a dominant series. So one is more dominant than the other and there's one series. Or it could be some are codominant and some are recessive compared to everything else. So it really depends on the question here. You need to pay attention. Alright, second last, gene interactions. The gene interactions is quite interesting because there are two genes. It's sort of like dihybrid, but it's not because it only influences one phenotype only. In dihybrid, two genes, two alleles each, but two types of traits. So tall, short, purple, white. But in gene interactions, two genes influence one phenotype only. For example, we learn the example of chicken feathers, either colored or white. That's just one phenotype, one type of thing. Okay, the F2 ratio again depends on the gene and organism, but it tends to be somewhere around 9:3:1, but like a modification of 9:1:3:1. I'm just going to write here, modified 9:3:1. So it could be 9 plus 3 to 3 and 1, depends on the situation. So you need to read the question carefully. Now, last but not least, we have autosomal linkage, which is the idea that two genes are on the same chromosome. So they are more likely to be inherited together as they are considered linked genes. Now, don't be confused. Autosomal linkage is different from when we say autosomal recessive or autosomal dominant. Autosomal linkage refers to, again, linked genes. It means genes that are on the same chromosome. Now, the F2 ratio will depend on which alleles are on the same chromosome, which alleles, which alleles are on the same chromosome. That's pretty much it. Okay, so yeah, that's the summary so far. And with that, we are ready to go into mutations. Now, mutations is something you have already learned in chapter 6 in AS, but now we're going to add on more examples to that, and also link it to the patterns of inheritance we already learned. So let's go into a little recap about chapter 6 first, and then I will tell you what diseases you should remember for the exam, right? So yeah, here's the chapter 6 recap. Now, gene mutations is defined as random change in DNA sequence during DNA replication. Usually, it's caused by mutagens. For example, ionizing radiation, UV radiation, or maybe mustard gas. And these mutations, random change in DNA sequence, actually cause altered codons in mRNA sequence, and of course alter the amino acid sequence of the polypeptide chain. Now, just a reminder, every time we talk about mutations or describe mutations, we always describe what happens at every level. So what happens at the DNA level, then what happens at the mRNA level, and then what happens at the polypeptide level. So it's the same in A2, same idea, same sort of pattern in mark scheme as well. Okay, so mutations, what is the purpose? So it actually is not all bad. Sometimes it can result in new alleles. A base or two change can result in a new phenotype. Again, alleles, what we mean by that is different form of one gene. And of course, it affects the phenotype. I already said that. Moving on. Now, gene mutations, we usually talk about this in terms of single base mutations. Okay, so we're not talking about large scale changes, we're just talking about very minuscule level gene mutations. So there are three types here. We learn substitution, insertion or addition, and deletion. Insertion and addition is the same thing, by the way. So again, substitution, you should know this already. It's like just changing the base. Insertion is like there's one extra base now, and deletion is one base is removed. Again, according to slides, we do learn only a change of one nucleotide. We learn the simplest form of mutations, and that is called point mutation or base mutation. Now, in base substitution, so the substitution of one base, only one nucleotide is replaced by another. Okay, it could result in three types of mutation: a silent mutation, a nonsense mutation, or a missense mutation. Now, I'm not going to teach this in detail. I'm just going to screen through, okay, really quickly, because you already know this. This is recap, and you already took the AS exam, right? Okay, perfect. So, base substitution again, silent mutation means there is the triplet code has changed, but the amino acid is unaffected because just so happen the mRNA codon codes for the same amino acids. Nonsense is when stop codon is introduced, and this causes an incomplete non-functional polypeptide. Basically, the polypeptide is gone, it's not useful anymore. Missense is when a different amino acid is incorporated, and therefore this might change the shape of the polypeptide, resulting in maybe for it to not to function as well as before. Yeah, so it may be still functional, but in worst case scenario cannot lah. Okay. Alright, how about base insertion and deletion? Now, insertion and deletion I put together because you know it has a very similar effect. And the idea is, number one, it will cause a frame shift. It may cause a frame shift. No, wait. It will definitely cause a frame shift. Deleting or inserting one nucleotide of DNA will change which bases are read together. So it pushes it back or brings it forward, and either way, that's not a good idea. You can see how the frame has shifted here. And this results in all subsequent amino acids being affected. So everything after the mutation, all the bases, all the triplet codes are affected, all the codons are affected, and therefore all the amino acids affected, and this protein is definitely faulty and non-functional. Now, the second situation this could result in is because of the frameshift mutation. Maybe a stop codon is accidentally introduced, and obviously this also forms a non-functional polypeptide. Okay, with that little recap, now we're ready to go into 5 important disease examples. Number 1 is sickle cell anemia. Number 2 is albinism. Number 3 is haemophilia. Number 4, Huntington's disease. And number 5 is cystic fibrosis. You can see here that I've listed the pattern of inheritance they take on. we will go through them one by one. Just so you know, about 3 of these diseases have specific symbols that represent alleles of the gene, and this is not usually given to you. Okay, sometimes given to you, but sometimes not, and you do need to know how to write them already in the first place. Okay. Whereas with other diseases like cystic fibrosis and albinism, it might not have the specific symbol, so you can use whatever alphabet you want, as long as you write this sentence in the front defining what those alleles are. Let maybe big A be normal and small a be the disease allele, so on and so forth. So make sure you define it if you are deciding. If they give you, then you don't need to define it, okay? Alright, moving on to the first one, sickle cell anemia. Yay, classic example. You already learned this, so let's just breeze through this, right? Sickle cell anemia is an inherited blood disorder. It affects the structure of hemoglobin, and the cause is really a base substitution in gene coding for beta globin, which is one of the polypeptides in the hemoglobin, right? Hemoglobin is made up of 2 alpha globin and 2 beta globin. So it's a problem there. Now, where is the mutation? The mutation is in the gene coding for beta globin, right? And CTT actually became CAT, and therefore this changed the mRNA codon and therefore changed the polypeptide, resulting in different 6 amino acid of the polypeptide chain, altering the primary structure, and then therefore influencing the secondary, tertiary, and quaternary structure. So it becomes a faulty protein. Now, mutated beta globin causes hemoglobin molecules become less soluble in low partial pressure of oxygen. Okay, and therefore, if it's less soluble, it's going to stick together. It's going to form long fibers, because you can imagine stick and then pull away. It's going to form long fibers. Cells become crescent or sickle shape as a result. That's why it's called sickle cell anemia. And as a result, also beta globin carry less oxygen. Now, the problem here is that sickled red blood cells break down faster as well and cause lack of red blood cells. It's very often that a sickle cell gets stuck in capillaries like this and block blood flow as well. So this is called a sickle cell crisis. So it might cause like pain or like a numbness or soreness in certain regions of person with sickle cell anemia. But it's not all bad. It has one good thing about it, that is that it gives protection against malaria and plasmodium infection, and this we will learn more in the next chapter. Okay, why does it give a protection against malaria? So we'll hear about sickle cell anemia again. Now, right now, again, this chapter is about inherited change. So let's go straight into what causes sickle cell anemia and what the genetics are. So HbA is how we represent the normal beta globin allele. HbS is how we represent mutant beta globin allele. And obviously, if they are homozygous for the normal one, they are normal. The person's normal. If they're homozygous for the sickle cell allele, then obviously the person has sickle cell anemia. But what if one normal allele is the person has one normal allele and one mutant allele? Well, the person becomes a carrier, but the person also has a sickle cell trait. Now, it's very interesting here because this person will have half the red blood cells which are normal. Okay, and half the hemoglobin is normal, but half of the hemoglobin is not normal, is problematic. So this person would have the sickle cell trait, but the person has enough normal hemoglobin to be able to function normally and not experience any symptoms. This is because both of these are codominant, both these alleles are codominant, so the carriers have the sickle cell trait, but again, doesn't have the disease exactly. Very interesting. So you can see a little pedigree diagram here. Okay, this is codominant, and this gene is on an autosome. so it is on not a sex chromosome. So you can see the pattern of inheritance here when a person who is completely unaffected pairs with a person who is definitely very affected. You realize that all the children are heterozygous, and therefore all the children have the sickle cell trait. They are carriers. And when carriers mate together, marry each other, whatever, and have kids, you realize that there is a chance that the kiddo has the sickle cell anemia. What are the chances? Well, you can draw a genetic diagram and figure it out, but I can tell you now from like a lot of practice that the probability of crossing two heterozygous organisms together, okay, and getting a homozygous recessive phenotype, genotype is 1/4, right? Don't believe me? Draw a genetic diagram. Anyways, that is sickle cell anemia. Moving on to albinism. So, albinism is caused by an autosomal recessive mutation. So it's not codominant, it's autosomal recessive. Again, what is autosomal recessive? Autosomal means the gene is not on the sex chromosome, it's on a normal chromosome, an autosome, and the allele that carries this problem, albinism, is recessive. This is a situation where melanin, okay, is missing from the eyes, the skin, the hair, and this causes pale white skin and hair and pink eyes, very interesting, and increased susceptibility to sunburn or skin cancer, sometimes even poor vision, and some behavior to cope, I suppose, because the person may avoid bright light. So very interesting. But the main symptom here is pale skin and hair and pink eyes. You do need to know the symptoms for the exams. Sometimes they do ask, okay? Anyways, what causes the mutation, what causes the disease actually, as we said, is an autosomal recessive mutation, specifically in this gene called TYR, see italics, right? And this gene codes for the enzyme thyroxinase. Thyroxinase is an enzyme, it's a protein, yeah? And this thyroxinase is involved in the conversion of thyroxine to dopa, and dopa is some sort of neurotransmitter which is needed for melanin production in melanocytes. Okay, there is a pathway here you can see, and you can see thyroxinase is involved not in one step but 2 steps of the way. Now, in general, we need to know that thyroxinase is an oxidase enzyme. It has 2 copper atoms. It binds to 1 oxygen molecule, and it is actually a transmembrane protein found at the membrane of melanosomes, which are large specialized organelles in melanocytes. Okay, story short, this is the melanocyte. Okay, this is a melanosome, and this is thyroxinase. Okay, so mutation of this particular protein here at the membrane of melanosomes in melanocytes would cause absence or inactive thyroxinase. And if you don't have this, you don't have all of these, and therefore an absence of melanin completely, right, in the person. Crazy. So very important to realize that it's autosomal recessive. This means that the trait is uncommon, yeah, and the person would need two recessive alleles in order to be albino. Alright, disease number 3. Disease number 3 is haemophilia. Haemophilia is a sex-linked disease. Specifically, it is caused by X-linked recessive mutations. This means that the haemophilia allele here is X small h. And this actually causes factor 8 to be not produced. Factor 8 is involved in the clotting of blood. So if it's not produced, this obviously reduces the clotting of blood. Now this is quite dangerous because any cut, a small cut of wound, internal or external, can cause excessive bleeding. It can cause large bruises, and if there's internal damage, the person could experience bleeding into joints or internal bleeding. So quite dangerous, yeah. I know a friend who has it, but did you know that the haemophilia is actually present in the British monarch? Crazy. Well, actually, not the current monarch, but it is in the family somewhere. Okay, the people you know are here, right? There's Queen Elizabeth. Can you see? I think you can see, right? Let me zoom. Can I zoom? Ooh, I can zoom. Right, you can see here that Queen Elizabeth is here, Prince Philip is here. You have Prince Charles and Prince Anne, Prince Andrew, Prince Edward, and under Prince Charles here, Charles is the heir to the throne. You have William and what's his name? There's only two sons. There's two sons, yeah, under Charles. But anyways, those fellas don't have haemophilia, but all the way up here, Queen Victoria had the haemophilia allele from somewhere. I'm not sure where, but she had it. She was a carrier. And you can see that it passed through generations, and interestingly enough, okay, passed to a lot of males. So you can see this generation here, because it's X-linked recessive, right? X-linked recessive, it is more likely to show up in males, especially when the mom is a carrier. Okay, why is that the case? Because again, in males, you only need one hemophilic allele in order to for it to be expressed. So you can see here, right, it's male affected. The genotype is X, small h, Y, right? Only need one recessive allele in order for it to be affected. Males can never be carriers because they only have one X chromosome. Okay, it's either normal or affected. Whereas in females, it's very likely that they become the carrier if they have a mom who is a carrier as well. Or the person could be unaffected entirely, so normal. So all carriers must be female. Again, a male with a carrier mother has a very high chance, a 50% chance of having haemophilia, which actually explains this generation of males getting haemophilia. Poor guys. But that's haemophilia for you. Now we are going to disease number 4. Yay! This is Huntington's disease. Now, unlike the previous examples, this is an autosomal dominant disease. It's caused by an autosomal dominant mutated allele. It's a neurological disorder, i.e. neurons will die, and people usually don't know they have it, or they don't experience any symptoms until middle age, around 28 to 65 years old. And this mutation, okay, is at the Huntington gene, or the HTT gene on chromosome number 4. Okay, again, it is not on the sex chromosome, it's chromosome number 4 here, and it's a dominant allele, meaning that it will express even if you just have one copy of the allele. Now the symptoms here when the neurons start to degenerate is involuntary movements, uninhibited motor control. So imagine a lot of trembling, a lot of not being able to pick up things, or just not working. You know, the hands are not doing what you want to do, your legs as well. Mood changes could be a thing, progressive mental deterioration, so memory loss, things like that. Brain cells might be lost as well. So yeah, it is quite tragic really. I shouldn't be so happy, but I'm trying to keep you awake and keep the pace going. I feel like this is a good pace, don't you think? Anyways, as we said, it is an autosomal dominant disease. The mutation is at Huntington gene. The normal recessive allele has around 10 to 35 repeats of this CAG, so this is base nucleotide bases in the DNA. The normal one is just 10 to 35 times, but in the Huntington gene allele, there is more than 36 repeats, more equals to 36 repeats of CAG. And somehow this of course this affects the glutamine, okay, CAG codes for glutamine, the number of glutamines inside the resulting protein, and causing the protein to misfold and of course malfunction. Now, the more repeats, okay, so you might have 36, you might have 40, you might have 50, right? The more repeats of CAG, scientists realize that the onsets are earlier. So if it has many times over, the onset means even the age where you experience symptoms might be even in babies. So that's crazy. Okay, so the longer, the more repeats of CAG they are, the more serious, the shorter the lifespan of the person. So because of the nature of autosomal dominant alleles, okay, you only need one autosomal allele. You only need one allele, one dominant allele, in order to express that phenotype. So most of the people with HD are heterozygous, okay, and it's very unlikely that a person with HD is homozygous, because that requires the crossing of two heterozygous parents at least, okay. And if the person is alive, then can pass lah, and two, they marry each other. But usually, as we said, the onset begins in the middle age, which is the marrying age. So it's very unlikely that they pass it to the child. But if they do, okay, you can see here, this means that they probably married young and raised a child younger before the disease hit. So yeah. Alright, let's go to disease number 5 now. 5. So this is the last disease. This is cystic fibrosis. Cystic fibrosis is not supposed to be taught in this chapter, supposed to be taught later, but it's also a inherited genetic degree. So I thought, why not? Why not teach it now, recap later? Makes sense. Okay, so number 5, inherited genetic degrees is due to a autosomal recessive allele of the CFTR gene. The CFTR gene stands for cystic fibrosis transferase. Hang on, I just checked it. So cystic fibrosis what again? Cystic fibrosis transmembrane conductance regulator. Man, that is quite a mouthful. Good thing you don't need to memorize that. You just need to know CFTR. Anyways, the CFTR gene normally, so the dominant allele, will code for a transmembrane protein in the cell surface membrane, and this acts as a chloride channel. The chloride channel actually has a binding site for ATP. I know it's a channel, so it's not supposed to, have active transport, but it's actually not, right? It's actually gated, but it's ATP gated, so it will only open if ATP is present. So it's still passive transport, okay? Facilitated diffusion. It's just gated by ATP. Anyways, what happens is that in these cells, usually lining the respiratory tract, right? You can imagine cell like this, right? The membrane protein is here. This is a CFTR protein. The channel, I don't know what's going on there. Okay, and what happens is chloride ions move out, and chloride ions actually cause water to move out by osmosis. And therefore, the mucus, which is secreted by mucus glands, by goblet cells, they they sort of get mixed up with this water and has a consistent texture. It's not too viscous, it's quite normal. But, and of course it's easily removed by cilia. But in a faulty CFTR allele, there's a base deletion. Okay, that's the most often one, or maybe a base substitution, causing an incomplete CFTR. And this results in a faulty CFTR protein. Now, when it's problematic, this in this cell, then these channels don't work anymore, so chloride ions don't move out anymore, and therefore if chloride don't move out, then H2O sort of stays inside and doesn't actually diffuse out that much. So less water leaves cells, and therefore the mucus that's on the surface of these cells, they form a thick, sticky consistency, and therefore this might cause problems because it cannot be removed by cilia that easily. So a person with cystic fibrosis will always have thick and sticky mucus produced at lungs, and therefore mucus is not effectively removed like we talked about just now. The mucus will accumulate, the mucus will trap bacteria and cause more infections. This really sounds like what? Chronic bronchitis, except this is a genetic problem. Now, because of more infections and more scarring, there might be reduced gaseous exchange. Also, there's a longer diffusion pathway because there's just so much mucus stuck in there. And very often a person with CF, that's the short form for cystic fibrosis, have difficulty breathing, wheezing. Very often they have to have a ventilator. There's a lot of coughing as well because it's a normal response to try to clear that mucus, and this causes the lungs to be quite scarred, as we mentioned just now. Well, CFTR proteins actually not only at the respiratory pathway, but also at the pancreatic ducts. So this might be a problem because reduced digestion, damage of pancreatic tissues may cause diabetes. So in addition to their wheezing and breathing difficulties, they may need to take some enzymes, okay, like orally in order to help with their digestion. The last one is quite sad as well, blocked sperm ducts or oviducts, because you do know that our reproduction organs do have some mucus secretion, right? So everywhere with mucus will be affected here, and in this case, for sperm ducts and oviducts, this will result in reduced fertility. Just a reminder again, cystic fibrosis is caused by an autosomal recessive mutation. This means that two parents that look symptomless, they can be carriers for the disease, for example. So in this case, let me represent it by big A, normal, small a, cystic fibrosis allele, faulty CFTR allele. This is more proper. And you can see that two carriers could result in a few kids who have cystic fibrosis because they may be homozygous recessive. However, if you have a carrier who married maybe a normal person, it's very likely, it's not likely at all that the kids, 0% that the kids will get cystic fibrosis. It is only possible that they become carriers as well. Now all these pedigree diagrams, you actually can stare at it longer, try to make sense of it, write genetic stuff on it in order to figure out the genotype just for practice. Okay, I've put that there for you to label and stuff as well. And with that, that's it. That's 5 different diseases you need to know. So far in part 3, you learned sex linkage, codominance, multiple alleles, gene interaction, autosomal linkage, all the special cases that are not really monohybrid or dihybrid cross-like, not really the normal dominant or recessive type of relationship. And then the last part of here, we learned about mutations. Now don't forget this particular chapter has 4 parts, but part 4 has been posted earlier, and part 4 is all about gene expression. So if you haven't watched the video yet, I encourage you to watch it and finish off this chapter and get it over with. Now, again, I want to say that this chapter is an understanding chapter, and even though I just went through some details really quickly, these details are quite, I mean, you still need to know them, but a lot of the questions that come out is not that straightforward. A lot of the questions that come out are new scenarios, and you will need to use your understanding, your conceptual understanding of what's going on with inheritance in order to solve the problem or solve the puzzle and answer the questions. So more past years, guys. Right, I'll see you next video. Bye bye.